and very warm welcome. Dear students, today I will be talking about the types of female gametophyte or embryo sac in angiosperms. In our previous lecture, we have studied various types of embryo sacs or female gametophyte, which were mainly of three types: the mono, bi or tetrasporic embryo sacs. The monosporic embryo sacs were further divided into two types, the bisporic were further divided into two types, and the tetrasporic embryo sacs were further divided into 7 major types. We have reached a Drusa type. Today we will be discussing Peperomia, Pinia, Drusa, Fritillaria, Plumbagella and Plumbago. Apart from them, there are some minor type variations within the normal type of embryo sacs. Now let us discuss Peperomia type. Campbell in 1899 and Johnson in 1900 reported that in Peperomia pellucida there are 2 mitotic divisions in the 4 nuclei of the cynommegaspore after taking up tetrapolar arrangement and result in a 16 nucleate gametophyte. According to Johnson, in the final organization at the micropylar end form the egg and a synergid, 8 fuse to form the secondary nucleus and the remaining 6 are cut off at the periphery of the embryo sac. Campbell also observed a similar type of embryo sac development, and according to him the egg apparatus is invariably 3-celled. Approximately 8 nucleate form the secondary nucleus and the remaining 4 to 6 nuclei are cut off as antipodal cells. Fagerlint in 1939 made a detailed study on Peperomia pellucida. He found that the outline of the cynommegaspore may be more or less spherical or pear-shaped, and in the later case there is slight protuberance at the micropylar end of the cynommegaspore shows 3-celled egg apparatus, 8 polar nuclei and 6 peripheral cells, and the embryo sac derived from the pear-shaped cynommegaspore. Johnson in 1914 reported a different type of embryo sac development in Peperomia hispidula at the 8 nucleate stage. 2 nuclei are seen at the micropylar end and 6 nuclei at the callosal end. At the 16 nucleate stage, 4 lie at the micropylar end and 12 at the callosal end. 2 nuclei of the micropylar group now form the egg and single synergid as in other species. But the remaining 2 nuclei of the micropylar group and all the remaining 12 secondary nuclei, and there are no antipodals. As this is different from Peperomia pellucida, it is recognized as a subset, namely Peperomia hispidula type. Murthy in 1959, who studied several species of Peperomia, stated that in the mature embryo sac the egg apparatus may be 2 or 3-celled and there may be 6 to 9 polar nuclei and 4 to 6 peripheral or antipodal cells. Maheshwari in 1963 opined that in view of such variability it was not necessary to maintain the 2 subtypes Peperomia hispidula and Peperomia pellucida types. Now let us discuss Pinia type. Stephens in 1909 described this type of embryo sac development in 3 genera of Piniaceae, namely Pinia, Brachysiphon and Sarcocola. Here the nuclei of cynommegaspore take up tetrapolar arrangement and divide twice to form a 16 nucleate embryo sac. The 16 nuclei are arranged crosswise, 3 nuclei of each quarter later cut off as cells, while the 4th remains free and moves to the center. There are thus 4 triads and 4 polar nuclei. Generally, the central cell of the micropylar triad alone functions as the egg. The central cell of the other triads also simulate the egg. This type of embryo sac development is found characteristic of the family Penaceae and Euphorbiaceae. Maheshwari and Johari in 1941 studied the embryo sac development in Acalypha indica, which is almost similar to Penia type but differs slightly in the organization of mature embryo sac. Here each peripheral group consists of 2 cells and 2 nuclei of each quarter remains free and migrate to the center and fuse to form the secondary nucleus. Thus there are 4 groups of the 2 cells and at the periphery 8 free nuclei in the center in this type of variation. Now let us discuss Plumbago type. Hopt in 1934 reported this type of embryo sac development in Plumbago capensis. The 4 nuclei of the cynomagaspore are arranged in a crosswise fashion. They undergo another division resulting in 8 free nuclei arranged in 4 pairs. A nucleus of the micropylar pair is now cut off to form the lenticular egg cell. Of the remaining 7 nuclei, 4, probably 1 member from each of the 4 pairs, undergo a slight increase in size and gradually approach one another and function as polar nuclei. The remaining 3 nuclei degenerate occasionally, 1, 2 and all 3 of them are cut off at the periphery to form cells which persist and assume an egg-like appearance. Synergids are entirely absent. The Plumbago type also occurs in Plumbago coccina and Plumbago scandens. This type was also found in 2 genera of Plumbaginaceae, namely Ceratostigma and Vogelia, and is not known outside this family. Now let us discuss Drusa type. Hakanson in 1923 in Drusa oppositifolia of Umbellifereae recorded a 16 nucleate embryo sac of different nature. After the 2 meiotic divisions were over, the 4 nuclei of the cynomagaspore take up 1 plus 3 arrangement, and this is followed by 2 plus 6 arrangement and then 4 plus 12 nucleate stage. The 4 micropylar nuclei give rise to an egg apparatus and the upper polar nucleus, and the 12 clausal nuclei to a lower polar nucleus and 11 antipodals. Later, this type of development was also noticed in Melotus japonicus, Crucianilla latifolia, Smilacina stellata, etc. Sometimes due to the failure of division of some clausal nuclei at the 8 nucleate stage, there are sometimes fewer than 11 antipodals. The embryo sac of Mainthemum canadense also shows Drusa type, but is about 13% of the ovules. During the division following the 8 nucleate 2 plus 6 stage, the clausal spindle fuse in pairs, resulting in 6 diploid nuclei instead of 12 haploid nuclei. Now let us discuss Fritillaria type. This type was first described in Fritillaria, persica and lilium. The cynomagaspore in this type of embryo sac development takes up 1 plus 3 arrangement. The 3 clausal nuclei are present very close to each other. During the next division, the micropylar nucleus divides normally, but the 3 clausal spindles fuse to form a single common spindle, so that at the close of the division there are 2 haploid nuclei at the micropylar end and 2 triploid nuclei at the clausal end. One more division occurs, resulting in 8 nuclei, of which the 4 clausal micropylar and are haploid. The mature embryo sac finally consists of an egg apparatus of which all the cells are haploid, the 3 triploid antipodals and a tetraploid secondary nucleus formed by the fusion of 2 polar nuclei, one haploid and the other triploid. The fusion of 3 clausal nuclei of the cynomagaspore during the division was first observed by Bambiconi in 1928, hence the fusion process is known as Bambiconi effect. Cooper in 1935 found the Bambiconi effect and the formation of Fritillaria type embryo sac development in the entire tribe Lilioidae and several other genera belonging to diverse families like Piper, Tamarix, Cornus, Rudbeckia, Gaillardia, Tulipa, Clintonia, etc. The fusion of the 3 clausal megaspore nuclei may take place when they are either in the prophase stage or in early metaphase. In the former case, the secondary 4 nucleate stage is processed by a secondary 4 nucleate 1 and the subsequent sequence then is as follows: megaspore mother cell, primary 2 nucleate stage, primary 4 nucleate stage, secondary 2 nucleate stage, secondary 4 nucleate and lastly the 8 nucleate stage. Normally, all the 4 megaspore nuclei are of same size, but in some plants in the micropyle nucleus is larger than the rest, in which case the nuclei of the secondary 2 and 4 nucleate stage show no appreciable difference in size in spite of their triploid nature. Rarely the clausal nuclei are smaller than the micropylar, although they are triploid. Sometimes the basal nuclei of the secondary 4 nucleate stage fails to divide as a result of 6 to 7 nucleate gametophytes are formed in some species of Statica and Gagia. Depending on this, the mature gametophyte has either 1 or 2 antipodals. Now, let us discuss Plumbagella type. This is a reduced Fritillaria type of embryo sac and is reported only in Plumbagella micrantha. The 4 megaspore nuclei take up 1+3 arrangement. A large vacuole separates the 3 clausal nuclei from the micropylar. The 3 clausal nuclei eventually fuse to form a single triploid nucleus and results in secondary 2 nucleate stage. This is followed by a secondary 4 nucleate stage in which the 2 micropylar nuclei are haploid and the 2 clausal are triploid. There are no further divisions. The nucleus nearest the micropylar end organizes into egg. The triploid nucleus nearest the clausal end organizes into a single antipodal cell, and the remaining nuclei, one haploid and the other triploid, fuse to form a tetraploid secondary nucleus. This mode of development is similar to the Fritillaria type, and the only difference is that in Plumbagella the development is arrested at the secondary 4 nucleate stage and the 4th division is omitted. Now, let us discuss aberrant and unclassified types of embryo sac. In addition to the 7 fairly distinct and well established types of tetrasporic embryo sacs, there are few which appear to be more or less isolated. Of this, the embryo sac development in Chrysanthemum cinerariaefolium is special and interesting. The embryo sac development is sometimes somewhat peculiar in this taxon and was reported by Martinelli in 1939. The embryo sac is tetrasporic and the megaspore nuclei take up 1+2+1 arrangement, so that there is 1 nucleus at each pole and 2 nuclei in the middle. The two central nuclei become separated from the terminal nuclei by vacuoles and may either fuse to form a single diploid nucleus or may simply remain close to each other without undergoing any fusion. Based on the behavior of the two central nuclei, there are two variations within this. First variation is the two central nuclei fuse to form a diploid nucleus. After fusion, all the three nuclei divide twice to form the three groups of 4 nuclei each. From the micropolar quarter are produced the egg apparatus and the upper polar nucleus, all haploid. The clausal quarter gives rise to 4 antipodals, all of which are haploid. The central quarter is comprised of diploid nuclei. One of these functions as the lower polar nucleus and the remaining 3 are organized as additional antipodals. Occasionally, fewer than 12 nuclei are found either because of failure of some divisions at the clausal end or because the central diploid nucleus undergoes only one division instead of 2. Now let us discuss the second variation. In the second of the two alternatives, that is the two central megaspore nuclei do not fuse but only lie in contact with each other and function directly as the polar nuclei. The micropolar as well as the clausal megaspore nuclei divide twice, forming 4 nuclei at each pole. Thus the embryo sac is 10 nucleate. All the 4 nuclei form the clausal quarter organized as antipodals. Of the micropolar quarter, 3 nuclei organize into egg apparatus, while the 4th one along with 2 central nuclei function as polar nuclei. However, there is no regularity in the behavior of clausal nuclei. Sometimes there is only one division or none, and based on that the mature female gametophyte is 9 or 7 nucleate. Occurrence of more than one type of embryo sac development in the same species of or even in the same individuals is common in some cases. Kapil and Prakash in 1966 in the Delosperma cooperi reported 14% ovules showing polygonum type of embryo sac, 18% endymion type and 68% various tetrasporic types. Similarly, AB in 1971 recorded allium 31%, adoxa 53% and polygonum 16% type of embryo sacs in orchis patens. But the interesting thing was that the mature embryo sac contained only 6 nuclei in all of them. In the general, like Chrysanthemum, erigeron and euphorbia, the development of megagametophyte may be mono, bi or tetrasporic. The environmental conditions to some extent can influence the type of embryo sac development in some cases. Hagelmist and Grazi in 1965 have shown that the temperature influences the type of embryo sac. For example, Ulmus glabra show adoxa and Drusa type of embryo sac in addition to some rare types. According to them, low temperature 15 to 19 degrees centigrade considerably enhanced the frequency of 8 nucleate adoxa type of embryo sacs. They explained that the temperature affects nuclear division more than the cell growth and vacuolation. At low temperature, the nuclear division is delayed in relation to cell growth, so that the central vacuole appears after meiosis 1, separating the 2 megaspore nuclei to the end of the megaspore embryo sac. By a single mitosis after meiosis 2, an 8 nucleate adoxa type of embryo sac is formed. At high temperature, the nuclei divide rapidly in relation to cell growth, so that the vacuolation appears only after the second meiotic division, separating a single nucleus at the micropolar end from 3 at the clausal end. By 2 more divisions, a 16 nucleate embryo sac of the Drusa type is formed. In capsicum, it was reported that the embryo sac formed in winter were bisporic and those formed in summer were of the monosporic type. Thus, the environmental factors like the temperature can influence the type of embryo sac development. With this, we conclude our today's topic. Thank you.